Good morning, dear students. Welcome to grade 10 biology lesson 6. Well, today we're going to discuss about active mechanism. Before going to start our topic, let's have a quick review of our previous lesson. In previous lesson, we talked about transport of materials through the plasma membrane. Means how the material like ions, atoms, and some other molecules, how they enter inside of the cell through plasma membrane or cell membrane. So this movement of these atoms, ions, or molecules occurs in two methods. One is passive mechanism and second one is active mechanism. In passive mechanism, the molecules move from an area of high concentration to an area of low concentration without using any energy. Passive mechanism, the movement of molecules from high concentration to low concentration area without using any energy. So this passive mechanism can take place in both living and non-living cells. This passive mechanism occurs in three forms. One is diffusion. Three forms. One is diffusion. Second one is facilitated diffusion, and third one is osmosis. So in diffusion, in a normal way, the movement of ions, atoms, or molecules from regions of higher concentration to regions of lower concentration, this process we call as diffusion. The other time I give some examples like perfume bottle, and if you drop ink, like a drop of ink in water, slowly the ink concentration, like from high concentration to low concentration, the ink molecules will spread throughout the cup. In the similar way, I gave one more example, like a tea bag. When you drop a tea bag in hot water, you can see the concentration of the tea slowly will spread all over the cup, like in the water. So means here they are not using any energy. The second type of this passive mechanism is facilitated diffusion. So here in a facilitated diffusion, they use these proteins. Like in a previous lessons, I show you here in a facilitated diffusion. You can see on the screen, like even in our previous lesson, in a facilitated diffusion, they use these carrier proteins to like the entering of these molecules from outer environment to inside of the cell. They use these carrier molecules like carrier proteins. So by using of these carrier proteins, ions or molecules or atoms can enter from outer environment to like inner environment of the cell. So this type of the diffusion we call as facilitated. So you have to remember, in a facilitated diffusion, atoms, molecules, or ions, they use these carrier proteins. So here also they are not using any energy. So now on the screen you are seeing this facilitated diffusion. They are using the carrier proteins. So by using the carrier proteins, these atoms, molecules, and ions enter inside of cell. Next, the third type of passive mechanism is osmosis. So here, osmosis is a quite different process from diffusion. Here, the molecules move from low concentration area to the area of high concentration area of high concentration through cell membrane, or we already know that it's a semi-permeable membrane. Once again, I am repeating, osmosis is a quite different process from diffusion. In this process, molecules, especially water molecules, they move from lower concentration area to high concentration area through semi-permeable membrane. So here also, for this movement, these molecules or ions or atoms, they cannot use any energy. So without using energy by these three methods, the molecules will enter from outer environment of cell to inner environment of the cell. Means they will enter from outside to the inside of the cell by through this cell membrane. So well, today we're gonna talk about the other type of mechanism, that is active mechanism. So here in the active mechanism, atoms or molecules or ions, they will use energy. You can see this topic in the page number 68 in your material. So there is the topic active mechanism. In the active mechanism, cells use energy and they use carrier proteins to transport material in or out of cytoplasm. You already know about the structure of the plasma membrane. So this is a lipid bilayer model or bilipid layer model. In this bilipid layer model, there is lipid molecules. So between the lipid molecules, there is some proteins which we call as carrier proteins. So in active mechanism, cells use energy and sometimes they use carrier proteins to transport material in or out of the cytoplasm. So here, passive mechanism occurs both in living and non-living cells, but when it comes about active mechanism, so this active mechanism only occurs in living cells. This active mechanism does not occur in non-living cells because this active mechanism they use the need energy. Okay, so here in active mechanism, in active transfer, the movement of molecules from area of low concentration to area of high concentration, similar like osmosis. But what's the difference between the passive osmosis and active osmosis? The passive osmosis, they don't use the energy. And in active osmosis, like in the active mechanism, they use energy, ATP, to transport materials from outside of the cell into inside of the cell. At the same time, they will use the same energy to excrete the cell waste from inside to out. Okay, in active transport, let's have a look in the picture. So in active transport, a carrier protein on membrane binds with molecule that will enter in the cell. Like earlier, what I said, in active transport, these carrier proteins on the membrane, like in a cell membrane, binds with the molecule that will enter the cell. Once the carrier protein inside the cell, this carrier protein detach or leave the molecule using energy from ATP. You know already know that most of the cells, all living cells, they use the energy in the form of ATP. Means, let's imagine there is a plasma membrane. In the plasma membrane, there are some active proteins. Okay, these active proteins binds with the molecule, whatever the molecule. For example, let's say sugar molecule, or let's say uh, sodium ion. So this sodium ion wants to enter inside of the cell. So how the sodium ion enters inside of the cell? So on the screen you are seeing. So here in a yellow color part, you are seeing that is a carrier protein. This is the carrier protein. So this carrier protein and these in a blue color, you are seeing these are the ions or molecules. So during this active transport, this uh, carrier protein attaches with the molecule, and once this carrier protein inside of the cell, like in the other part of the cell membrane, this carrier protein detaches, means they leave the molecule inside of the cell by using of energy. That energy is in the form of ATP. So that's why you must remember active transport need energy to transport these molecules or ions or atoms. So here there is some excuses, like as the concentration of the target molecule increases, the difference between the conditions inside and outside of the cell facilitate the transport of further molecules. So finally, you have to remember for the active transport, the need carrier protein and energy. Without energy, they cannot, like there is no active transport. So in the second paragraph, you can see, sorry, in the third paragraph, in the page number 38, both energy and carrier proteins are needed for active transport. If the activity of this carrier slows or ceases, or if ATP cannot be generated, there is no possibility of active transport. Once there is no possibility of the active transport, cell will die because the cell needs these ions and molecules for their several metabolic activities. So once the lack of these ions or atoms or molecules, cell will die. So what are the exceptions? What are the reasons? There are some reasons for the lack of this active transport. The active transport, like there are some reasons slowdown of active transport. There is if the carrier proteins are slowed down or seizes, the carrier proteins cannot function, or there is another more reason if these carrier proteins cannot use this ATP. In these both cases, the cell will die because they cannot transport these ions, minerals, ions, atoms, and molecules. So now, so there is a good example to show this active transport. So on the screen, so you are seeing a picture of this picture explains sodium potassium pump. We call as sodium potassium pump. You can see the same picture on your textbook on the page number 38. There is a figure 1.37 mechanism of active transport. So in that picture, this picture gives the information of how the ions like sodium ions and potassium ions will enter and go out of the cell by using this mechanism we call as sodium potassium pump mechanism. So well, so the active transport of sodium and potassium ions through the plasma membrane. Normally, sodium ions are concentrated on outside of plasma membrane, while the potassium ions are concentrated on the inside. Let's imagine there is a cell. The cell is surrounded by the plasma membrane. There is a cell outside environment and inside environment. So this sodium potassium pump explains how the ions will transport from outer environment to inner environment by using active mechanism. Sodium ions are concentrated on the outside of the plasma membrane, while the potassium ions are concentrated on the inside. So this concentration difference leads the movement of both sodium and potassium ions. Let's say, let me give an example. So like earlier I told you, the sodium ions are concentrated on outside of the plasma membrane and potassium ions are concentrated inside of the plasma membrane, like inside of the cell. So to enter these potassium ions, so first these carrier proteins catch some potassium ions from inside of the cell, and they will go to the outside environment, and they will leave these potassium ions. As they leave the potassium ions, right away these sodium ions attach to the active transport proteins, attach to the transport proteins, and right away these transport proteins bring these sodium ions inside of the cell like an exchange. So for example, let's say two ions of potassium on the screen. You can see on the screen. So two ions of potassium go out from inside to the sorry. It's quite different from the explanation. On the picture you are seeing, let's say here on the picture you can see that there is a concentration of the potassium ions outside, and the concentration of the sodium ions inside, isn't it? So here, to enter these potassium ions from extracellular environment to inside of the cytoplasm, some potassium ions should go out to maintain the balance. So once some sodium ions will go out, and right away these transport proteins or carrier proteins catch these sodium ions, and it will bring this potassium ions inside of the cell, like an exchange. If, for example, let me give an example. In a room, there is 10 number of the students. The room is very small. Or let's take another example. Just imagine the lift. Okay. So in a lift or in an elevator, there are 5 members. The capacity of the elevator is also 5 members. But there is a one person came from outside. He has a meeting urgently. He wants to use the elevator, but the capacity of the elevator is only 5. So to enter this person into the elevator, one person should go out, isn't it? So if the one person should go out from the elevator, this person, the X person, can enter in the elevator, then he will attend the meeting or work. In the same way, so here inside of the, on the picture, what you are seeing, inside of the cell, there is a concentration of sodium ions, and outside of the cell, there is a concentration of potassium ions. So here, to enter the potassium ions inside of the cell, there should be some sodium ions should go out from the cell. So in this process, to move these ions, they use the carrier transport. First, this carrier transport catches this some sodium ions and bring it outside of the cell. Once the sodium ions get outside of the cell, right away these potassium ions attach to the this carrier protein, same carrier proteins, and these carrier proteins bring these potassium ions inside of the cell. So once the potassium, these carrier proteins bring the potassium ions, so to detach, to separate these carrier proteins and potassiums, they will use the energy. So this energy is in the form of ATP. So this is the best example which explains the active transport of like active mechanism. So you must remember sodium potassium pump model explains the movement of active transport, how the molecules or atoms can enter and go out of the cell. So this is about the active mechanism. Well, next, let's go to the page number 39. There you can see the words endocytosis. So here, this is another process. It's quite different from this mechanism. So this process also explains how some bigger molecules can enter inside of the cell or into the cytoplasm. Endocytosis is taking of large molecules into the cytoplasm. So here, endocytosis only occurs in the case of the size of the molecules are quite bigger, means only these larger molecules enter in the process of endocytosis. So here, There is the three types of the endocytosis. One is phagocytosis, or we call as cell eating, and second one is pinocytosis. It's also known as cell drinking, and the third one is receptor mediated endocytosis. You must remember, endocytosis is another process. It's quite different from the active and passive mechanism. So, in the active passive mechanisms, only the smaller molecules like ions, atoms, and some small molecules can enter by the process of passive and active mechanism. When comes about the bigger molecules, like large molecules, they enter in the different process we call as endocytosis. So in the endocytosis, like you can read, endocytosis is the taking of large molecules into the cytoplasm. So the size of molecule is an important factor in this method. Movement in or out of cell means only the large molecules, okay, can enter or go out from the by the process we call as uh, endocytosis, because these larger molecules they cannot enter through the these pores of the cell membrane. So now these three types of the endocytosis, one is phagocytosis, pinocytosis, receptor mediated endocytosis. Okay, let's talk about phagocytosis, or we call as cell eating. So phagocytosis is the taking of solid large particles into the cytoplasm. So this is a process, the taking of solid large particles into the cytoplasm. It without concern this active and passive mechanism. So this is a good example, like microbes, like virus and some bacteria, like they will enter in the body of leukocytes by the process of phagocytosis to kill these foreign particles. And then the same way in amoeba, the food enter into the food vacuole by the process of phagocytosis. We call as cell eating. Then the second one is pinocytosis, also called as cell drinking, taking of the large dissolved particles by a food vacuole into the cell is named as pinocytosis. For example, let's say in digestion process, when the food is get digested, the small intestine absorb these food molecules, like digested like food molecules, like nutrients. So this from this small intestine, they have the finger-like projections we call as villi. When the villi is absorbing these nutrients, so these nutrients are dissolved in the water. So when the villi are absorbing this water, at the same time, the molecules, like some bigger size molecules which are dissolved in the water, they will enter inside of the villi. Then later on, this villi send these nutrients and water into the blood. So this kind of the process we call as cell drinking. Cell drinking means the molecules which were dissolved in the water. So this type of the absorption we call as pinocytosis. Then the third one is receptor mediating endocytosis. So taking of specific molecules into a cell by inward budding of membranous vesicles that have receptor proteins specific to the molecule being taken. So here in this process, so this membrane, the plasma membrane, produce some vesicles like a small buds. So these small buds will take this large size like molecules and bring it into the inside of the cell. So means in special mediated endocytosis, the cell membrane produce a small membranous vesicles. So these membranous vesicles get these large size particles and bring it into the inside of the cytoplasm. So especially this cholesterol taken into the cell by this method, receptor mediated endocytosis. So apart from this endocytosis, there is another method we call as the exocytosis. Exocytosis is nothing but when a cell produce this waste, when a cell produces the waste, so this waste will go out from the cell. So this process we call as exocytosis. The process by which the cells remove large particles from the cytoplasm is called exocytosis. The process by which cells remove large particles from the cytoplasm is called exocytosis. Any waste substance present inside of the cell will go out by the process of exocytosis. So this is the lesson today, and before that you must remember endocytosis has three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. In a phagocytosis, we call as cell eating. So in cell eating, so the big size or solid large particles enter inside of the cell. We call as phagocytosis. And when comes about the pinocytosis, dissolved large particles enter along with the water inside of the cell. We call as cell drinking. And the receptor mediated endocytosis is, so these large particles enter inside of the cell. So as the cell membrane produce a special vesicles like a structures like a buds. These special vesicles bring these large particles inside of the cell. So these are the three types of endocytosis, and the exocytosis is nothing but the cells remove the large particles from the cytoplasm, is which is called as exocytosis. So this is the end of the lesson, and this is also end of our chapter, that is the cell micro universe. In the next for next chapter, I will upload or I will send a file in the either through Fedena or through WhatsApp. I will send this PDF of the second chapter. So we can continue with the second chapter in the next one. Thank you very much. Have a good day. See you in next class.